Good morning. Good morning, everyone. Happy Monday morning. If you could just type your first name or your nickname into the chat for me, I'd appreciate that. All right, very good. All right. So we have a sort of an unusual week this week. We're only meeting for lab today, and we're going to be talking about the control of microbial growth using chemical compounds. You have 2 exams this week, which is why we're not meeting for lab on Wednesday or Friday. You have a lecture exam on Wednesday, and you have a lab practical exam on Friday this week. So your other work is less this week. You have 1 lecture topic, which is the antimicrobials, the antimicrobial drugs. No quiz for that. And you have 1 lab this week, which is what we'll be doing today. No lab homework this week. Okay, so it's a very unusual week. Hold on just a second. I can't quite get this this light correct. All right, see if that works a little better. All right, so if you just come in, go ahead and type your name or your nickname into the chat for me. And we'll go ahead and get started. I'm going to pull up our schedule just so we can take a look and see what's coming up. So again, this week, and you should be seeing that on your screen right now, you should be seeing our syllabus, the schedule at the end of our syllabus. Here's where we are right now on March 15th. So you have this 1 lecture topic this week, which is the antimicrobial drugs, 1 laboratory topic, which is controlling the growth of microbes with chemicals. And then you have your 2 exams, lecture exam on Wednesday, lab exam on Friday. And just like last time, those exams will be open and available to you during the day, and you'll just need to get them to me by 8 o'clock. 8 o'clock on Wednesday night for the lecture exam, 8 o'clock on Friday night for the lab exam. Now looking ahead to next week, it's another busy week for us. You have 2 lecture topics about metabolism. In lab, we're going to be talking specifically about how microbes use carbohydrates. We'll look at carbohydrate metabolic pathways and also fermentation. And of course, next week is the time that your first draft of your pathogen assignment is due. So don't forget that. Make sure that's up on your schedule, that those questions that you need to answer for the pathogen assignment are due a week from Wednesday, March 24th. I will go ahead and pull up our Blackboard page again. Here we are. You should be looking at our Blackboard page right now. Again, this says the week of March 15th. Here's your to-do list. Again, it looks very long because you have instructions for the lecture exam here. You have instructions for the practical here. You've had lecture exams before. This is your 3rd lecture exam. It's not going to be any different from the other ones. And your 2nd practical exam, very similar to your 1st one. Okay, so that's up on Blackboard for you if you have any questions. Okay, so I'm going to go ahead now and open up our lab slides for today about controlling the growth of microbes with chemicals. And that's what you should be seeing on your screen now, the very first slide for this lab exercise. So our objectives for today are to look at 4 very common families of disinfectant chemicals. So we won't be looking at individual products so much as we'll be looking at the families of chemicals that products belong to. We'll compare the relative effectiveness of different chemicals. And importantly, we'll talk about the limitations of these chemicals. You'll hear me say that it's really important that we understand disinfectant chemicals because we have to know what their limitations are, when it's appropriate to use them and when it's not appropriate to use them. The other thing we'll be talking about today is how we test new chemicals for their effectiveness as being a disinfectant. So we have some vocabulary to review today. Remember, there's a difference between, say, a sanitizer or a cleaner and a disinfectant. And there's another type of chemical called an antiseptic, which is very similar to a disinfectant, but is used very differently. The key word for us today is disinfectant. When we talk about a disinfectant, we're talking about a chemical that can eliminate common pathogens from inanimate objects and surfaces. So we're not talking about using disinfectants on living tissue. We're going to use them on surfaces and objects in our environment in order to kill and get rid of any pathogens that might be on those materials. Now, an antiseptic is very similar because it will also eliminate common pathogens, but it's not applied to inanimate things. It's not applied to surfaces and objects. It's applied to living tissue and primarily to skin. So whenever we apply a substance, a chemical substance to our skin for the purposes of killing microbes, that is what we refer to as an antiseptic. There's a couple of other terms that we need to be familiar with, and these are discussed further in the lecture this week. One is this term bactericidal and bacteriostatic. You'll hear these words used when people talk about antimicrobial drugs, antibiotics, and so on. When we discuss a chemical, whether it's a disinfectant or an antibiotic, and we call it bactericidal, we're saying that it can kill bacteria outright. In comparison, if a chemical is described as bacteriostatic, what we're saying is that it prevents that bacterium from growing anymore. It prevents it from dividing and creating new cells, but it doesn't kill it outright. We have a fair number of antibiotics and other antimicrobial drugs that are bacteriostatic. They're not actually going to kill bacteria outright. When you tell students that, they get confused because it doesn't make sense that the drug doesn't kill these bacteria. It just stops them from dividing, but they're still in your body. And the reason those drugs are effective is because it keeps the number of bacterial cells in your body very low, and it allows your immune system to kill them. Oftentimes, what makes us so very sick when we have a bacterial infection is the fact that those bacteria rapidly multiply and they sort of get to the point where the immune system can't handle them. But if we keep the number of cells low with a bacteriostatic drug, the immune system can get on top of the infection and eliminate it. So yes, there are drugs that will kill bacteria outright, but there are other drugs that just prevent them from dividing long enough that the immune system can step in and eliminate the infection. The truth of the matter is that whether you're using a bactericidal or a bacteriostatic antibiotic, it's your immune system ultimately that's getting rid of the infection. The drugs help your immune system clear the infection. We learned this, unfortunately, we learned this reality during the AIDS epidemic. We discovered that if you don't have a functioning immune system, all the antibiotics in the world aren't going to save you from a bacterial infection. If you don't have a functioning immune system, and remember, people who have AIDS, they don't have a functioning immune system anymore. And those people, when they get a bacterial infection, they succumb to it. They die from it because even though you can give them antibiotics, their immune system is ultimately what's needed to get rid of the infection. So yeah, these drugs are very important to us in medicine, very important because they're very helpful for the immune system, but you've got to have a healthy immune system, a functioning immune system in order to fully recover from infections. All right, I'll get off my little side tangent there. Couple of things to remember about disinfectant chemicals. No single chemical is best. I often get asked, as a person who teaches microbiology, I often get asked, what should I buy to disinfect in my house? What's the best disinfectant to clean my house with? And the answer is there is no one single best. There are best chemicals to use in different situations, depending on what you're trying to disinfect and what microbes you think you might be dealing with in your environment. You have to know the chemicals because you have to be able to match the chemical to both the microbes you expect might be lurking in your environment and the conditions in that environment. The material that, whatever it is you're trying to disinfect, the material that it's made out of matters. The pH of that environment or that material, how soluble the disinfectant is. Does it dissolve into whatever it is you're trying to disinfect, or does it crystallize out? Solubility matters. The toxicity of the chemical matters, right? I mean, we could kill microbes in our environment using very, very toxic things, but we don't because we get exposed to those disinfectants and we don't want to harm ourselves. The other thing that really plays a role is this last one, the amount of organic matter that's present in the place that we're trying to disinfect. In other words, how dirty is the area that you're trying to disinfect? Is it caked with food or caked with mud or soil? Is there a lot of material present on the surface that you're trying to disinfect? If there is, you really need to get rid of it first and then disinfect. So a chemical can be really effective in one situation under one set of circumstances and be really ineffective in another. So we have to know what they can do and what their limitations are. So I mentioned at the beginning of the lab today that there are tests we can do to evaluate disinfectant chemicals in terms of how good they are, again, in certain situations. And one of the most common tests that gets run is what's called a DRT, or a decimal reduction time. The other term that is used for this test is D-value, although in my experience, DRT is the more common name that you'll see. So this is defined as the time that it takes, and it's specifically in minutes, the time that it takes to kill 90% of the microbes in a particular spot, in a particular location. So that's the decimal reduction time. The time that it's going to take for that chemical to kill 90% of the microbes in that situation. On this slide, you're looking at a table that comes out of a textbook, and it provides DRT values for several different chemical compounds. So it shows us the name of the compound over in the left-hand column. It shows the bacteria that these chemicals were tested on, and you can see they were tested on 3 different kinds of bacteria. Shows you the concentration that they were used at, and that's, you know, the manufacturer determines what concentration you need to make it up to. And then there's some other statistics here, but this is the one we're looking at, the D value, or the DRT in minutes. So let's think about what this means. The first chemical in the table is hydrogen peroxide, and it's 10% hydrogen peroxide, which is the strength that it's typically used at when people use it as a disinfectant. And you'll see it has a pH of about 5. So against Pseudomonas aeruginosa, and if you remember, this is an organism that tends to live in our environments, even our man-made environments, and it's an opportunistic pathogen. It likes to get into the bodies of people who have a compromised immune system, and it can cause problems for them. So against Pseudomonas aeruginosa, the D value is 3.8 minutes. And that means that it takes 3.8 minutes of exposure to hydrogen peroxide for 90% of the Pseudomonas aeruginosa to be killed. So if I started off with 100 cells on my countertop and I applied some hydrogen peroxide, I would have to leave the hydrogen peroxide for 3.8 minutes in order to kill 90 of those cells. I'm not killing all of them, but I'm killing 90% of them at 3.8 minutes. Now you can see the numbers are different for different species, right? For Bacillus subtilis, I have to leave the hydrogen peroxide on longer. I have to leave it for 5.6 minutes. For Staph aureus, I have to leave it on for 8.7 minutes. One of the biggest misconceptions that people have with disinfectants is that you can spray them on a surface and then wipe them off, right? I mean, that's how most people use chemicals in their homes, right? You spray it on and then you wipe it off. In reality, if you're trying to use a chemical as a disinfectant, you need to spray it and leave it for a few minutes and then wipe it off. So a lot of times what people are doing when they think they're disinfecting is they're really just cleaning a little bit. They're wiping things off the counter, but they're not really disinfecting the counter. They may be reducing the number of pathogens on that counter, but they're not really disinfecting. Most people don't understand that chemicals often have to sit in order to work. All right. So you can see that there's all kinds, they actually use the same 3 organisms in this table, and they made comparisons for several different disinfectants. And you can see the DRT values, the D values really range broadly for the different chemicals and for the different microbes. So again, you have to know your chemicals. You have to know how to use them correctly, especially if you're in a healthcare facility. So we can calculate DRT. We can calculate it. We have some certain starting number of organisms that we use in the lab. We put the disinfectant on top, and then we test at intervals, usually minutes. and to look at how many cells are left. And when you hit 90% reduction, that's your DRT. That's the time it takes to get rid of 90% of the cells. There is another test that gets used to evaluate disinfectant chemicals. This one is called the Aoac test, or the Use Dilution Test. It gets its name because it's produced by the American Official Analytical Chemists, the AOAC, Use Dilution Test. Now, they tend to use, again, 3 different organisms for this test, but they're different from the ones you saw in that table. Pseudomonas is still here. Pseudomonas aeruginosa, that one can be a tough one to kill. Staphylococcus aureus is still here. But we also have Salmonella on the list. So this use dilution test is kind of interesting in the lab. What you do is you grow up pure cultures of these 3 organisms, and you drop these little stainless steel cylinders into the broth tube. And I'll show you this in just a minute. I'll show you what they look like. So you take these little metal cylinders, and they've been sterilized in the autoclave, and you drop one into each of the broth tubes, and then you lift it out and you let it dry. So essentially you've coated this metal cylinder with cells, and you're going to set those cylinders down and let the cells dry onto the cylinder. And then you're going to place that cylinder into the disinfectant for the recommended dilution and the recommended time period. You're going to take it back out, and you're going to place it into some fresh nutrient broth and see what grows. And by doing a little bit of math, you can determine how many cells were killed during that exposure to the disinfectant. Now, you can only test bactericidal products this way. You can only test products that kill cells outright, because you're going to be counting cells and which cells have lived and which have died. This is what those little cylinders look like. In this picture, you can see several Petri dishes sitting side by side, and they have some sterile filter paper sitting in the bottom. And you can see all these little metal, they look like little metal rings almost. These rings get sterilized in the autoclave, then they get dipped into cultures, pure cultures of bacteria, and then set down in these plates to dry. So they're basically coated in cells. And then you just put the cylinder down into a disinfectant for the recommended time and see how many of the cells got killed by taking the cylinder out and then putting it into some nutrient broth. Any cells that have survived the disinfectant can then grow in the nutrient broth. So those are the 2 primary testing procedures that you'll read about, and that will be referenced in these tables that are in textbooks telling you essentially how good a disinfectant is. And it allows us to compare the disinfectant chemicals based on these test results. So that if it's my job, for example, to clean the floors in a hospital facility, and I know I have to disinfect the floor, I can pick the correct chemical for that job based on, the kind of material the floor is made out of and how easy or how hard it is to disinfect that floor. Now, there are other tests available for comparing disinfectants. I've only shown you the 2 that are very common. For example, sometimes what scientists will do is they'll grow up a microbe in culture, they'll make a broth culture, and then they'll just directly put the disinfectant into the broth tube. And they'll see after a certain number of minutes that the manufacturer recommends, they'll see how many cells survive it. So that's possible to do too. It depends on what your needs are. It depends on whether you're trying to get a qualitative comparison of chemicals or a quantitative comparison. Remember, when we hear that word qualitative, that's just telling us whether or not something happens. So for example, I might open a textbook and I might see that, or I might open a manufacturer's reading material for a chemical, and I might see that it states that this chemical will disinfect if you apply it for, 3 minutes. That's different from saying this chemical will bring the number of cells down 90%. One of them is qualitative, whether you're saying if it will or if it won't, and one of them is quantitative. It's actually measuring a number. It's actually quantitating a number. So both of those types of tests are available to us in reference literature, and we have to be able to read those and know what they mean. All right. So before we start talking about the different families of disinfectants, let me ask if anybody has any questions so far. Any questions about what a disinfectant is? Remember, we said we gave it a definition that's pretty broad, right? We said it's going to eliminate common pathogens from inanimate objects, from surfaces and objects. So it's pretty broad. It's a very broad definition. So when I hear that something is a disinfectant, I have to take a step further and do a little more research to know how good a disinfectant it is. I might need to look up a DRT value to know exactly how to use it and when it eliminates 90% specifically of the microbes. All right, I'm going to jump back in. Let's start talking about these different families. Remember, I said there are 4 primary groups of disinfectant chemicals. The alcohols is number 1. The chlorinated compounds is number 2. Number 3 are a group called the iodophores. And number 4 is a group called the Quaternary ammonium compounds. So these are big groups of chemicals. Let's start with the alcohols. There are many different alcohols that are available to us commercially to use as disinfectants. One of the more common ones is isopropanol or isopropyl alcohol. This is what's often referred to as rubbing alcohol. You can buy this, in your CVS. You can buy little bottles of it. You can also buy great big gallon jugs of it if you're going to use it as a disinfectant. But any alcohol will have these properties that I'm going to talk about. So even, plain old ethanol will work as a disinfectant if it's used correctly. Now, alcohols have a really nice characteristic that I've listed right here. They are rapidly bactericidal at 60 to 90% solution. Remember when we talked about aseptic technique earlier in the semester, and we talked about cleaning off our workspaces, we said that you don't want to use alcohol full strength. It actually works better when it's diluted. So it needs to be somewhere between 60 and 90%, not full strength. The most common percentage that's used in laboratory spaces and healthcare facilities is 70%. Now, the water that's being used to dilute the alcohol actually makes the alcohol better. It makes the alcohol rapidly bactericidal. And that's really what we want from a disinfectant, right? We want it to work quickly so we don't have to stand around and wait. Now, I'm going to list a scope of activity for the alcohols and for the other families of disinfectants. And what I've done is I've written 4 specific types of organisms or other objects, that living things, in other words, that we might be trying to get rid of. So instead of just saying, oh, it's a disinfectant against bacteria, we're also going to ask, does it kill mycobacteria? And remember, mycobacteria is the type of bacteria that causes tuberculosis. Does it kill fungi? Does it kill fungal cells? Does it kill or destroy viral particles? And finally, is it effective against spores? So we will ask specifically these 4 questions for each one of our groups. When it comes to alcohol, alcohol will kill Mycobacterium. So, if you work in a facility, a hospital facility, or any other kind of long-term care facility, or a doctor's office type of facility, and you know that you see a fair number of patients with TB, then you have to assume that, your disinfectant is going to need to kill off the TB organism in the environment. So it's important to know if your disinfectant is up to that task. Remember, the Mycobacterium organism, and the genus is Mycobacterium. The species is tuberculosis. So it's called Mycobacterium tuberculosis, and it causes a disease that we call tuberculosis as well. Remember, it's a very unusual organism, right? It's different from other bacteria. It can't be gram stained. Remember that? We said that this is a kind of bacterium that secretes this waxy material around the outside of itself. That material is called mycolic acid, and it literally, it's wax. So you can't gram stain this organism because the stains can't get through the wax. If you try to gram stain it, you won't get any color on the cells. You just can't. So we have to use special stains on this cell, this organism. And we have to use special disinfectants sometimes. We have to know if our disinfectant is going to work because of that waxy material. All right. Alcohols will also kill fungal cells. They will destroy viral particles. The one thing alcohols can't do for us is get rid of spores. It will not destroy spores. Remember, spores are very hardy. They're not cells anymore. They've gone into this state of a sort of in between life and death. They're not metabolizing. They don't need nutrients. They don't secrete wastes when they're spores. And they're very resistant to destruction. So spores will not be destroyed with alcohol. So what exactly is alcohol doing to kill things? Well, it actually works on proteins, and it does a process that we refer to as denaturation. When you hear that word denature, what we're saying is it's going to cause that protein molecule to unfold. Remember, proteins have a shape. All proteins start off as a string of amino acids, all joined together. And the order of the amino acids determines the different proteins. There can be hundreds or even thousands of amino acids in that string. And once the string is made, once that strand is made, it folds into a 3-dimensional shape. And it's the shape of that protein that allows it to do its job. What alcohols do is unfold proteins, denature proteins, and they can no longer do their function anymore, and the cell dies. Now, alcohol has limitations like all disinfectants do. The primary problem with alcohol is this: it can damage material over time, specifically rubber, plastic, and certain kinds of lenses. So if you were to disinfect, for example, a rubber tube to a piece of equipment in your hospital, or some kind of plastic surface over and over and over again with alcohol, that material is going to degrade over time and it's going to have to be replaced. The other limitation to alcohol is that it's very expensive. A 1 gallon jug of pure alcohol, concentrated alcohol like this, probably runs about $100 now. So if you're buying this as a disinfectant, you have to take that cost into account. On this slide, you're looking at an image that depicts this process that we call denaturation. On the left-hand side, you're looking at a drawing of a protein in its molecular shape. You can see it's basically a green string, right? And that string is all twisted and turning. It has these helix loops in it. It's a 3-dimensional molecule. And it's built from a strand, a long string of amino acids. It's the way this thing folds that will give it its ability to do a job in the cell. So every single protein has a unique shape. That shape is critical because if it's not in the right shape, it can't do its job. When you expose protein to alcohol, this happens. It just unfolds it. It takes it back to its long strand of amino acids. So it's still a strand of amino acids. It's just not folded up anymore. So it can't do its job and the cell will die. This represents a spore. Remember that cells can sporulate in certain species of bacteria. Remember, we said the genus Bacillus and the genus Clostridium are capable of sporulating. And essentially what happens is the cell goes away. All that's left behind is this core of basically DNA and a couple of enzymes. And it's surrounded by layer after layer after layer of hard protective materials. And these materials are very resistant to destruction. So that DNA gets protected until we're ready for the cell to come back, until the conditions in the environment get better and the vegetative cell can come back. So alcohols are not going to be effective against spores. The next family of chemicals are chlorinated compounds, and the most common chlorinated compound is plain old bleach. So all bleach is, household bleach, all it is a chemical called hypochlorite. And obviously you can go right into any store and you can buy a jug of hypochlorite. You can buy a jug of household bleach. Bleach, like alcohol, is rapidly bactericidal, but you have to dilute it. And this time you have to dilute it a lot. We dilute bleach to 10% to get the best activity from it. So it's very diluted. Now here's our list for scope of activity. Bleach is effective against the organism that causes tuberculosis, but you have to leave it in contact with the surface or the object for minutes of time. In other words, it's not enough to spray the bleach on and then wipe it off. You got to spray the bleach and you got to let it sit for minutes and minutes of time if you're trying to kill TB. So, that could be a bit of a drawback depending on the institution that you're in and whether or not you're trying to get your disinfecting done quickly. So that's certainly a limitation, a drawback to bleach compared to alcohol. It will kill fungal cells. It will destroy viruses, but again, if you're trying to kill viruses, you got to let the bleach sit for minutes of time. Now, here's an interesting thing about bleach. It will destroy spores, but you have to use a higher concentration and you have to leave it again for minutes of exposure time. So you could argue that bleach is better than alcohol, right? Because bleach can destroy spores. But you could also argue that bleach is worse than alcohol because it takes a long exposure time to get things killed. Now, when it comes to the mechanism of action of bleach, what is it doing to kill bacteria? Well, believe it or not, we don't really know. We don't really understand everything that bleach does inside or on a bacterial cell. We do know this, though. When you mix bleach in water, you're going to get an acid formed. And we know that acids can oxidize cells. In other words, they can take electrons away from cells. And that, of course, will damage molecules in the cell and ultimately kill the cell. There are lots and lots of bleach products on the market. One of the biggest companies is called Clorox. You've probably seen all kinds of Clorox chemicals in the grocery store. You can buy all different combinations of chemicals for different purposes in the house. We have to remember, though, that bleach has a pretty long list of limitations. Sorry about that. Somebody came to my door and my dog is alerting me. So whenever you're using bleach in a pH, a basic pH, in other words, if the environment that you're trying to clean has a relatively high pH, it's going to be less effective in that environment. Remember, we know that bleach, when it's mixed with water, when it's diluted, becomes an acid. So if I apply it to a surface that has a high pH, a basic pH, the acid and the base are going to neutralize each other. We need the Clorox bleach or the other kind of bleach to stay as an acid. So it's important that we know what pH we're applying it to. If it gets neutralized, it's not going to work. We know that bleach is a lot less effective in the presence of organic matter. And remember, we said when I use this phrase, I'm talking about dirt, I'm talking about soil, I'm talking about old food stuck to things. You know, if you're in a restaurant or if you're in a cafeteria situation and you're trying to, let's say, disinfect a dishwasher, you got to make sure that there's no old food stuck to surfaces in the dishwasher because the bleach won't be able to get through that and disinfect the metal. We know that bleach solutions don't last very long either. So if I'm going to use bleach as a disinfectant, I have to make up fresh solutions each week. I have to make up a fresh 10% solution once a week. Because when you dilute bleach to 10%, over time, it's going to degrade in your bottle. So you've got to make it fresh. You don't have to do that with alcohol. You don't have to make your alcohol solutions up every week. You can keep using the same one. We know that bleach damages materials the same way alcohol does. It just damages different materials than alcohol does. Bleach is very damaging to metals. It can corrode them over time. It's also very damaging to fabrics, and you probably already knew that. If bleach gets on fabrics, it will take the color out of fabric. So you just have to be careful if you're going to be applying it around fabric. You don't want to get it on the fabric. If you put a concentrated solution of bleach onto fabric, it will eat a hole through the fabric. So again, it's very damaging to fabric. Here's one that you may or may not be familiar with. You have to be very careful not to mix bleach with ammonia. Now, we don't tend to use as much ammonia anymore as a cleaning chemical because it has a particularly bad smell, and most consumers don't like the smell of ammonia. It used to be much more popular than it is now. But there are still people who use ammonia for certain cleaning jobs in their home, especially. If you mix bleach with ammonia, you will produce chloramine. And chloramine is a toxic gas. Every year in the news, there's always at least one story of one homeowner who was either taken to the hospital or who was found dead because they mixed ammonia and bleach. You know, sometimes people think that if you have two chemicals and they're both good, if you mix them together, it'll be even better. And every once in a while, there'll be a story about somebody who was cleaning their bathroom and they mixed bleach and ammonia together, and it will produce this invisible toxic gas, and it will kill you if you don't get out of the cloud of gas quickly enough. So that's a pretty big drawback for bleach. You got to be very careful. Something else to know about bleach, which is not a limitation, it's actually something good. It's a lot cheaper than alcohol is. Remember, I said that a gallon of undiluted alcohol will run you about $100. A gallon of bleach will run you, $5 or $10. It's very inexpensive compared to alcohol. All right. The third of our groups of chemicals is the iodophore chemicals. Now, it's important to remember that these are not iodines. Iodine is a particular chemical. And when you take iodine and you mix it with what's called a solubilizing agent, you get an iodophore. All right. So betadine, for example, which is a really common brand name for iodophores. You may have seen it if you've ever worked in a hospital or a doctor's office or a long-term care facility. We often use betadine or products like it as antiseptics. We put them on the skin to clean the skin prior to surgery, prior to certain procedures being done, placement of catheters, things like that. So it's very commonly used as an antiseptic, and you can also use it as a disinfectant. The brand name Betadine is also available as this generic form, which is called povidone iodine. These are iodophores. Now, they are bactericidal, but they are not rapidly bactericidal. So they don't work as quickly as alcohol and bleach does. Scope of activity, iodophores will kill the organism that causes TB. They will kill fungi, they will kill viruses, and they will destroy spores. But you got to leave them on for a long time for both fungi and for spores. So that's important to know. So far, we've said both bleach and iodophores can destroy spores, but you've got to leave them on for minutes and minutes of time. Now, similar to alcohol, iodophores are going to damage protein by denaturing it, by unfolding it. But iodophores also damage DNA. They get in through the cell wall, they unfold proteins, and they damage DNA. Remember, we said there's a chemical called Graham's iodine, right? And we said Graham's iodine is used as a mordant. It helps drive stain through the cell wall of bacteria, right? Well, these iodophores have iodine in them. So the iodine really helps drive the disinfectant into the cell. Limitations of their use, believe it or not, if you let your iodophore solution sit on the shelf for too long without making it up fresh, some bacteria can actually start to grow in it. There are some species of bacteria that are just resistant to this stuff. So you have to make up your solutions fresh, similar to what we saw with the bleach. Iodophores are less damaging to materials than alcohols and bleach are, but there's one material that iodophores will destroy, and that's silicone tubing. We use a fair amount of silicone tubing in medicine. We use it for our machines that we use in hospitals and doctors' offices, and we use it for patient care. And iodophores will destroy it. This slide is just an image of what the povidone iodine or the other iodophore is doing when we spray it down onto a surface. This here in the middle of the drawing, this represents a piece of bacterial membrane. So you have to imagine that there's a big cell here, and we've cut off a little bit of the membrane so we can see what's going to happen. So this is the inside of the cell over here, and this would be the outside environment. And what they're showing us is that the iodophore, the chemical that they've got listed as PVP iodine, this is an iodophore chemical. What it's going to do is it's going to actually release this iodine. The iodine, like we said, can penetrate. It can penetrate through the cell wall. It can penetrate through the membrane of the cell. It's going to destroy DNA by damaging it. And it's also going to unfold or denature proteins, proteins that are in the membrane and proteins that are in the cell. The last family of chemicals that we're going to talk about today are the quaternary ammonium compounds. It's quite a name. They get that name because they're made out of an ammonium ion chemical. So that's NH4 plus, and what's called a modifying agent. So you can't just apply ammonium as a disinfectant because you're going to, it's going to bind with other ions and it will be inactivated. So they add this second chemical to it called a modifying agent, but it's the ammonium that is doing the disinfecting. Now you've probably heard about some of these. There's a brand name called Hibiclens. That's a quaternary ammonium. This is a very popular antiseptic, again, in hospitals and doctors' offices. The generic brand of Hibiclens is just called chlorhexidine. There are lots of different brands on the market. In long-term care facilities and hospitals, there's a chemical called Virex that's very popular for cleaning floors. That has a quaternary ammonium in it. Just like the other families that we talked about today, it is bactericidal, right? In order to be a disinfectant, it has to be bactericidal. But just like the iodophores, it's not rapidly bactericidal. So you have to keep that in mind. Only alcohol and bleach are rapidly bactericidal. It happens in seconds, not minutes. So that's why we choose alcohol or bleach as our disinfectant in the lab. It's still bactericidal, but you got to leave it on for longer periods of time to kill bacterial cells. Now, some important things to know: it is not effective against the organism that causes tuberculosis. It will kill fungi. It will kill viruses, but only enveloped viruses. Only enveloped viruses. And it is not effective against spores. So this one's a lot more limited than the other ones we saw. But if you're trying to kill bacteria and you're not particularly worried about the TB bacterium, then this is a decent chemical. The other thing to remember is that in human medicine, humans get enveloped viruses, right? Remember from our virology lecture that most animal viruses are enveloped viruses. It's the plant viruses that tend to be what we call naked viruses. So Hibiclens and other quaternary ammonium compounds, they only work against envelope viruses, but that's okay because that's the kind of viruses we get as humans. We're just animals, so it works pretty well for us. Now, mechanism of action for these compounds, they also unfold protein. That's a very common mechanism, right? They unfold protein, but they also disrupt membranes. So you can see that the reason that they're only effective against viruses that have an envelope, the envelope, remember, is made out of membrane. And part of what these chemicals do is break apart that membrane. That's part of what makes them effective. On this slide, you're looking at a drawing of a bacterial cell right here. And what the artist has done is taken a little piece of the membrane right here and blown it up for us to look at. This is that little piece of membrane. Here's the heads and the tails and the heads. And then out here, the red circles that you see, these are those ammonium ions. So when ammonium interacts with membrane, it does this. It pulls the phospholipids apart from one another and causes them to form these smaller structures. So it breaks the membrane down, and the cell can't survive without its membrane. Limitations of use for these? Well, they're less effective in hard water. Remember, there are regions of the country and places around the world where the water that we use for things like cleaning has more or less compounds in it. It has different types of ions in it and so forth. The hard water, that term refers to water that has a lot of different ions in it. And if you try to use quaternary ammonium compounds in hard water, the quaternary ammonium will get inactivated. Here's something very interesting. These are less effective when they are applied with cotton material. So I said a moment ago that we often use quaternary ammoniums as antiseptics as well as disinfectants. Hibiclens, for example, is used to clean your skin, so it's an antiseptic. In operating rooms, before a patient has a procedure, we've often used the betadine type iodophores to clean the skin. But we've also started using quaternary ammonium compounds like Hibiclens to clean the patient's skin before the surgeon makes an incision. But you got to know, if you're the one applying it, that you can't apply it with cotton gauze because it won't work as well. The cotton sort of inactivates it somehow. So you have to use a different material. You have to use more of a sponge material that's made out of something other than cotton, because the cotton will bind up the ammonium and inactivate it. It's kind of interesting. There's one other limitation for quaternary ammoniums that we need to know about, and that's this last one. People who use concentrated forms of quaternary ammoniums in their work, so think about, for example, janitorial staff in hospitals. They're the ones who are mixing up the concentrated chemical with water every day to clean floors and things like that. If you get exposed to the fumes of these chemicals over time, you can develop a sensitization to them. Now, what do I mean by that? In the lungs of people who breathe in the fumes of quaternary ammoniums, their airways just get thicker and thicker over time. That's what the artist is showing us over here. So instead of having a nice sort of narrow layer of tissue lining their airways, they have a much thickened layer of tissue. So these people develop almost like an asthma after they've been exposed to these chemicals over time. It doesn't happen to everybody. There are people who've been working, as janitorial staff, and every day they mix up the Virex and they mop the floors and nothing ever happens to them. But there are other people who will breathe in those fumes every day, and over time, they develop what looks a lot like asthma. So you have to be aware of that so that you can provide staff with protective coverings so they're not breathing in those fumes, because we just don't know who's going to be affected by it and who's not going to be affected by it over time. The general rule is that you give your staff, you give them some kind of protection, breathing protection, so they're not getting exposed to those fumes. All right. So that brings us to the end of our discussion of these 4 families. And remember, what you'll need to do as you're reviewing this material is make some kind of chart for yourself, comparing them. Make a chart that has a column of alcohol, and then a column for chlorinated compounds, and a column for the iodophores, and a column for quaternary ammonia, and compare them. Have a box for mechanism of action for each one, and have a box for scope of activity for each one, and then the limitations of each one. If I were preparing this material for a test, that's what I would do. Just put them into a chart so you can quickly make comparisons between them in terms of what they do and how effective they are and what their limitations are. Does anybody have any questions about these chemicals? I know a lot of you have worked in healthcare facilities. Some of you have worked in long-term care facilities. So I'm sure you've seen some of these chemicals at least. They're very common and they're important because they're what allow us to disinfect. And like I said, some of them we use also as antiseptics. We use iodophores as antiseptics. We use alcohol as an antiseptic, right? Last time you had blood drawn or a catheter put into your arm, I'm sure you remember they wiped off that area with a little alcohol swab or something, right? So a lot of them get used as antiseptics and disinfectants. They're really important to us. We need them in order to control microbes around us. But we've got to know what they're capable of and what they're not capable of. We have to know how to use them, and we have to know what their limitations are. Any questions? All right. Yaxi is asking a question about the pathogen project. So in the couple of minutes we have left, I'm going to answer that. She asked about the citations, the references. When you read the instruction sheet for the pathogen assignment, you're going to see that it instructs you for each question that you answer. It instructs you to find 3 sources for each question. So underneath each question, I want you to list those sources. Now, I'm not being particularly strict in this assignment about how I want you to cite your source material. In other words, I'm not asking that everybody use the same citation style because we're still under these pandemic conditions. I know a lot of you will be going to the internet for source material. That's fine. That's not a problem. You don't need to go track down academic journals for this particular assignment. So how you cite is not really all that important. You just want to make sure that you give me a complete list of the 3 sources that you use for each question in the assignment. And I need to be able to use your citation to find the material that you used. So if you went to a website, for example, and I list several websites in the instruction sheet that I recommend that you use. If you go to the CDC website, for example, because you're going to answer the questions about cholera, you need to tell me where on the website you went. What's the URL? What's the full web address for the pages that you used? Don't just send me to the CDC. Give me the whole citation. Remember, the whole reason that we cite sources when we do a research paper or when we do an experiment and we write it up for a journal, the whole reason that you list your source materials, citations for your source materials, is so people can go find what you used. So I need to be able to go directly to the place where you found your source material. That's what you need to provide me. 3 sources for each question. Okay? Does that answer your question? Does anybody else have questions? Good.
Did you I tried to go back to watch some of your recordings on the YouTube page.
Yeah.
And I was asking, did you delete those? Because I couldn't find the whole NEC profile.
No, nothing's been deleted. Was there a particular video you couldn't find? Do you remember?
No, I just typed in our CRN and nothing was popping up. It was just a whole bunch of other random videos. So I just wanted to check in.
Yeah, no, nothing has changed. So you maybe there was maybe it wasn't the correct address. I don't know. If you go onto our homepage though in Blackboard, you can copy the URL for our playlist and do a copy and paste and see if you can get to it that way. But yeah, nothing's changed in terms of that. So
Okay. Thank you.
you're welcome. Anything else? All right, so remember, you're done now in terms of labs for the week. There's no lab homework to complete this week. You do have 1 lecture topic to view about antibiotics, antimicrobials, but there's no lecture quiz this week. And all of that is because you have a lecture exam on Wednesday and you have a lab practical exam on Friday. Okay, so make sure your notes are in order because you're allowed to use your lecture notes for the lecture exam. You're allowed to use your lab notes for the lab exam. Okay, and if any questions come up between now and those exams, you can message me. Okay, and we can talk about things. Make sure both exams are on your calendar. Okay. All right. If there's no other questions, I'm going to let you go. Enjoy the rest of the day. And I will talk to you later.